Yeah. Now is all right. Now audible. Sound is coming. Good morning to everyone. Good morning, good morning, good morning. Very good morning to everyone. So let's start the today topic. And today we are going to talk about prokaryotic cell. Something we started in the last class itself. Prokaryotic cell. Right? So if we talk about prokaryotic cell, typical example is different type of bacteria. Bacteria is a typical example of prokaryotic cell. Got the idea? So if we talk about prokaryotic cell shape, so usually 4 different shape we observe in prokaryotic cell. First type is, you can say, about bacillus. Second shape is coccus. Third shape is vibrio. And fourth shape is spirillum. So what is the meaning of bacillus shape? Bacillus means rod shape. Rod shape bacteria or prokaryotic cell. If the bacteria look like this shape, we are saying it is bacillus. Coccus bacteria means spherical. A spherical bacteria is coccus bacteria. If we talk about the shape of a spherical bacteria, it look like a sphere. Got the idea? If we talk about comma shape, vibrio is what basically? Comma shape. Sign of punctuation, comma is there, it look like this. I'm making bigger one. It is comma shape. And a spirillum is a spiral shape. A spiral shape. Got the idea? So if we talk about a spirillum, it look like this. So bacteria whose structure is like a rod, it is called bacillus. Who look like a spherical structure is coccus. Who look comma shape is called vibrio, and who look like a spring is what basically a spirillum. So these are the 4 different shape of the bacteria. Got the idea? If we talk about the size, I already told about generalized size in the last class. Got the idea? If we talk about the size, so what you can say about smaller than eukaryotic cell, getting the things in it, and multiply faster, increase its number, what basically faster than eukaryotic cell. That line I also wrote in the last class. Did you got the idea? Now if we talk about example of size, you help me. I told in the last class, I just want to check how many of you able to recall mycoplasma. Tell me the size and tell me the size of a typical bacterial cell. Tell me the size of a typical bacterial cell. So what is the size of mycoplasma? 0.3 micrometer. Well done. And a typical bacterial cell, what is the size? It is 3 to 5 micrometer, we can also write. It may vary. Got the idea? So we have seen the shape of prokaryotic cell. We have seen the size of prokaryotic cell. We also discussed about usually a prokaryotic cell is smaller than a eukaryotic cell, but it can multiply faster. So if I ask a small question based on concept I taught you earlier, who have usually more surface area versus volume ratio, prokaryotic cell or eukaryotic cell? Answer it. Well done. Very good. Very good. Keep it up. Because we already studied, we proved it mathematically as well in the previous class. If a smaller organism, a smaller cell have more surface area versus volume ratio than a bigger cell or bigger organism. Is it clear? We proved it previously, definitely. So student who attended that class, of course, you are able to connect with it. It's all right? Now we are going to discuss about the structure of prokaryotic cell. A structure of prokaryotic cell. Right? So give your attention. Now I am going to draw the structure of prokaryotic cell, and best method is what? You just note down the notes. If you're not understanding anywhere, type the questions. A relevant question, ki sir, these you told na, I didn't understood. And wherever anything required to explain, at the same position, definitely I will explain. Anything which required to explain later on, I will explain later on. Because in the very beginning, I already told you, it is not possible to fix every problem at the same time. Otherwise, we can't move on the chapter. Getting the idea or not? So definitely at the required time, definitely what you asked, I just put here. Okay, these are the questions asked by the students, and definitely we will cater it, we will try to answer it, we will definitely answer it when we find the right time for that. Okay, now that concept is implementing, that is going to answer. Like in this one, after 10 minutes, your many question is going to answer. You people asked me, ki sir, if prokaryotic cell don't have mitochondria, then how do you do the respiration? I think these are the questions of you people. Got the idea or not? You people also ask, sir, if prokaryotic cell don't have chloroplast, how they do the photosynthesis? So these are the questions is going to cater today. You asked 2-3 days back. It may be possible you disappointed yourself that time. Yeah, I ask a question to sir, sir not answer right now. So every particular explanation, definitely we are doing in a particular place when we find it is required at that moment. Got the idea? Now I am going to talk about the structure of a prokaryotic cell. Give your attention. So if we talk about a prokaryotic cell, I am just drawing a structure of a prokaryotic cell. Wait a minute. Suppose that this is a prokaryotic cell. So it is one of the covering. It is another covering. Boundary you can say, or covering you can say. These are, I am making one more layer of the membrane. Right. So a typical prokaryotic cell surrounded by how many membrane? 3 membrane. Got the idea? 3 layer. So if we talk about outermost layer, it is called glycocalyx. Outermost layer of a prokaryotic cell is called glycocalyx. Getting the things or not? Second layer from the outside is called cell wall. And the innermost layer is called cell membrane. Cell membrane or plasma membrane. Cell membrane or plasma membrane is the innermost layer. Got the idea? You just see in this innermost layer, I just made some enfolded structure. Na? Are you able to find it? This is an enfolding. So this enfolding of the plasma membrane which is present in the innermost layer is called mesosome. This is mesosome. Got the idea? Is it clear? One student is writing, sir, video is not clear. Is it for all of you? All of you are facing the clarity issue of the video, or it is okay for most of you? Is this clear? So please, dear, check your network. Because most of the student writing, yes, it is clear, means if it is not clear, means it is an issue of internet from your side. Is it all right? So please check your video, or you can do just going to reduce the quality of video. So your data loss will be less relatively. Got the idea? Is it done? So what I am saying here, this is an enfolding of the plasma membrane called mesosome, and here you look, there is another enfolding. Two type of enfolding I made, and this enfolding of the plasma membrane is called chromatophore. Got the idea? So two type of enfolding we have seen in the plasma membrane. One is mesosome, another is in chromatophore. Now we are going to draw the other structure which is present in the prokaryotic cell. In the center of the prokaryotic cell, you will find, I am just making like, it is a double-stranded extracircular DNA. Got the idea? I will don't say the name. I am expecting you will say the name. It is I just given a hint here. It is a double membranous circular DNA. Double membranous circular DNA, naked DNA, single circular DNA. What the name should I give for it? Very good. Well done. This is nucleoid. This is nucleoid. How many nucleoid is present in a prokaryotic cell? One. Many times this question came in the medical entrance exam, NEET exam. They ask a question, how many chromosome? They wrote chromosome, actually in true sense it is not true chromosome, but examiner asked question like this, how many chromosome present in bacterial genome or prokaryotic cell genome? Bacterial genome. Option is 2, second option is 1, third is variable from bacteria to bacteria, bacteria to bacteria, and if we just go for fourth option, it is 46. So what the answer of this question you will give? How many DNA or chromosome, anything people can ask, chromosome, here chromosome, actual true chromosome is not there, here who is there? DNA is there. So here we are writing in place of chromosome in bracket DNA because true sense chromosome not present in bacteria. I will tell you later on what is chromosome. So what is the correct answer will come? It is 1. It is 1. Always people struggle in the basics. Always people struggle in the basic. And when you go in a big interview also, people ask you basics things. Got the idea? Right? So how many? 1. If anybody going to ask you how many chromosome DNA present in bacterial genome, what answer will you give? You have to give 1 answer. Got the idea? Right. Now I am going to tell you, just give your attention. Have a look here. So this is the nucleoid. Beside of nucleoid in bacterial cell, extra genetic material is there. It is also a circular DNA. Nucleoid is what? Main genetic material. Main genetic material. Don't forget it. Now you can say, what is genetic material, sir? Genetic material who are responsible for transfer of characters in the next generation. Getting the things or not? Main genetic material, and who have a role in even the survival of the cells or organism. So main genetic material, or you can say main genome, or you can say genome. Genomic DNA. Nucleoid is what basically? Genomic DNA. Now beside of this genomic DNA, if this genomic DNA get disturbed, who may die? Bacteria may die. It is very important for survival of which one? Bacteria. If this DNA is not there in the bacteria, they can't survive. Not present in the prokaryotic cell because this is the DNA controlling all activity inside the bacteria. Are you getting the idea? Beside of this, extra genetic material is present. It is called plasmid. It is called plasmid. Plasmid is what basically? Extra genetic material. Extra genetic material. It is not the main genome, it is the extra genetic material. Got the idea? If a bacteria plasmid is not working or not present, it not happen, bacteria will die. It help in some of the function, but if this function even not occur by plasmid, bacteria survival is not going to impact it. So it is also, plasmid is also double-stranded circular DNA. It is also double-stranded circular DNA. And this plasmid is smaller than main genome of bacteria. This plasmid is, I am just writing, smaller than main genome. It is smaller than nucleoid. Smaller than nucleoid. So you got a basic idea, basic difference between main genome and extra genome. It is compulsory for survival of the bacterial cell. If it is not there, it is doing some additional work, helping work. If these plasmid is there, bacteria get certain help. What help it get, I will tell you later. Got the idea? But you please tell me, are you able to understand a basic difference between nucleoid or plasmid or not? I just want to fix it first. Are you very comfortable to differentiate between nucleoid or plasmid? Very good. Very good. Well done. So nucleoid is the main genetic material, or it is also called genomic DNA. If this nucleoid is not there in the cell or it get disturbed, survival of the prokaryotic cell will came on halt. Got the idea? Is it clear? I will come on chromatophore. Don't worry. One by one, I am coming on everything. I will come on mesosome as well. Got the idea? And these plasmid is extra genetic material. It is not compulsory. Plasmid is present in which one? Bacteria. Some bacteria is also there where plasmid is not present. But who is compulsorily present in a bacteria? Nucleoid. So, nucleoid you can say about universally present in prokaryotic cell. Universally present in all prokaryotic cell. Means no any prokaryotic cell or bacterial cell is there who have an exception of nucleoid. The nucleoid is not present. But plasmid, if we talk about, usually present. Mostly present but not compulsorily present. In most of the bacteria plasmid is there, but you cannot say in every bacteria compulsorily plasmid is there. Are you getting the idea? So you got the differences, a basic idea between nucleoid and which one? Now I am going to tell you, inside the plasma membrane it is a fluid filled matrix. This is fluid filled matrix, and you better know this fluid filled matrix is called cytoplasm. This fluid filled matrix is called cytoplasm. Got the idea? Inside the cytoplasm, who is present? One thing nucleoid is present, another thing plasmid is present, another thing which is present I am drawing. This is the ribosome, it is present also. Who is present also? Ribosome present also. So this is ribosome, this is another ribosome, this is another ribosome. Got the idea? And if many ribosomes join with a mRNA, it is mRNA. And it is ribosome. Got the idea? So mRNA plus ribosome all together it is called polysome or polyribosome or polyribosome. Got the idea? Is it clear in the mind? Free ribosome is also present. It is not necessary every ribosome is attached with which one? mRNA itself. Getting the things or not? When a ribosome attached with many ribosome attached with mRNA, messenger RNA, it is a type of RNA. I will teach you later. It is called polyribosome. Beside of that, in a bacterial cell or prokaryotic cell, some storage granules are present. I will decode everything, don't worry. Some storage granules are present, and these storage granules are called inclusion bodies. These inclusion bodies are what? Basically storage granules. It stores the material. I will talk about it. Storage granules. Got the idea? So these are the typical structure of a bacterial cell. And beside of that, on the bacterial cell surface, some extension is coming out. I am drawing that also. Suppose that from here a extension came out. This extension is called flagella. This extension is called flagella, and some another type of extension also arise from the surface. This is a tubular extension. This tubular extension is called pili. Another tubular extension are there. I am just drawing here. Another tubular extension, that is a hair-like projection. present all over the surface of body of the bacteria. These projections are called fimbriae. Fimbriae. So three type of outgrowth is there from the bacterial surface. One is flagella, another is pili, another is fimbriae. So you got the basic structure. I will give the explanation of every one. Don't worry about it. Now next I am going to discuss everything one by one. First you just see, I am just going to once again giving a brief about the bacterial structure or prokaryotic cell structure. Why I am saying bacterial structure? Because bacteria is a typical representative of a prokaryotic cell. So if you talk a typical prokaryotic cell, it look like this. Got the idea? Is it? So just have a look. I told a typical prokaryotic cell or bacteria surrounded by how many different layers? 3 layers. Outermost layer is called Glycocalyx. Second layer from outside is called cell wall. Innermost layer is called cell membrane. Got the idea? Clear? Clear? So you tell me, outermost layer. What is the name of outermost layer of a prokaryotic cell? Glycocalyx. Second layer from outside, cell wall. Well done. Innermost layer, cell membrane. Inside the cell membrane, fluid filled matrix is there. Agree na? Inside the cell membrane, fluid filled matrix is there. That is called cytoplasm. Got the idea? Well done, very good. Inside the cytoplasm, who is present? Nucleoid. Who is present? Plasmid. Who is present? Polysome. Who is present? Free ribosome. And who is present? Inclusion bodies. Agrees or not? So how many things are present? Any one is membrane bounded structure. Inside the cytoplasm, you see all the things, either nucleoid, or it is plasmid, or it is what? Polysome, or it is what? Inclusion body. No one is membrane bounded structure. And that is the reason while teaching the difference between prokaryotic and eukaryotic cell, I told you one thing. And what was that one thing? In prokaryotic cell, membrane bounded structure is absent. If it is ribosome, can I ask which type of ribosome is there? I am writing here ribosome. 70S is there or 80S is there? Tell me, help me out. Ribosome, which type of ribosome present in the prokaryotic cell? Please carry on. Very good, well done. It is 70S type of ribosome is there. 70S type of ribosome is there. Got the idea? What is 70S type? We will talk later when we will discuss about ribosome. What is 50S? What is 30S? Why 50 plus 30 not 80? Why we call 70? Many questions may be in your mind. Got the idea? But trust me, I will answer definitely later on. Got the idea? Got the idea? Right. Now I am going to tell about in inner membrane, that is the plasma membrane, there are 2 type of enfolding are there. One type of enfolding is called mesosome. Another type of enfolding is called chromatophore. 2 type of enfoldings are there. Got the idea? And from the bacterial surface, how many type of extension came? How many type of extension came? 3 type of extension came. What are the name of these extension? Please pin your response. What is the name of extension? Very good, pili. Second type of extension, flagella. Third type of extension, fimbriae. Got the idea? So a picture of bacterial cell or prokaryotic cell came in your mind or not? Tell me honestly. Did you got the idea? Now our duty is what? To explain everyone one by one. Ki what is this glycocalyx, what function done by this glycocalyx, what is the chemical composition of this glycocalyx. Got the idea? Is it clear? Now we are moving about one by one. Now you know what is present. Done? Clear? Now we are moving ahead. Give your attention. So first of all, I am going to talk about glycocalyx. Everything I wrote, I will explain. So it is outermost boundary of most bacterial cell. Bacterial cell, why I am writing bacterial cell? Because bacterial cell is an example of prokaryotic cell. And as I wrote most word, it means it is not present in all. If I wrote one student asking, can you please once differentiate between plasmid and nucleoid? Dear, you wait, I will once again write about this. So there I will say once the differentiation, don't lose your heart, be patient. I will once again definitely say the difference between nucleoid and plasmid. Don't worry, don't worry. I will write it also, na? Okay. I will write it also. I will write about plasmid, I will write about nucleoid, I will write about everything. I will give you the written proof as well. Got the idea? So listen it carefully. Give your attention, please. Okay, so have a look here. So glycocalyx, you understood? It is the outermost boundary present in most bacterial cell. If we talk about chemical composition, got the idea? So it is made up of, you can say, polysaccharide. Even it is made up of polypeptide. It depends upon the environment where bacteria is present. Either polysaccharide or polypeptide is there. Got the idea? Right? Now we are going to talk about glycocalyx, that is the outermost layer. That is the outermost layer. It may be of two types. If it is tough, it is called, or if it is loose, let me write loose first. If it is loosely arranged layer, glycocalyx have material chemically arranged loosely. Loosely, it is loose, you can simply write. It is called slime layer. Slime layer. And if it is tough, it is called capsule. Capsule. So the bacteria where capsule is there is called capsulated bacteria. Where capsule is not there, called non-capsulated bacteria. Got the idea? Now we are going to talk about function. So if it is a boundary layer, first function will come what? Protection. Second function is hold moisture and prevent bacteria from getting dry. From getting dry. Got the idea? Other functions, if we talk about the glycocalyx, is capsule lated bacteria protect itself from getting engulfed by WBC. Means they prevent themselves from phagocytosis. Protect themselves, protect the bacteria, better to write, from phagocytosis. Do you know this term? Phagocytosis. I am just giving you an idea. Detailed idea will be given by zoology sir when they will teach you a particular chapter, human health and disease. There they will give you the better. I am giving you a basic picture. Suppose that a bacteria enter in your body. Got the idea? A bacteria enter in your body. So in your body, who is there? WBC is a very warrior of your body. What WBC is doing? They are eating the bacteria. Cell eating is called. When a cell is eating the bacteria or anything unwanted coming in the cell is called what? Basically phagocytosis. It is engulfing of bacteria. They consume the bacteria, they digest the bacteria. So bacteria are not able to infect you. Got the idea? So capsulated or non-capsulated bacteria which have capsule. Bacteria which have capsule is called capsulated. Is called capsulated bacteria, and bacteria which not have capsule is called non-capsulated bacteria. So who protect themselves from our WBC? Capsulated bacteria or encapsulated bacteria? Who protect themselves? Who protect themselves from WBC attack? Capsulated bacteria protect themselves. Got the idea? Is it clear? So our infection chances getting diseased by a bacteria, who are more dangerous for us? Capsulated bacteria or non-capsulated bacteria? Who is more dangerous for us? Capsulated bacteria. Got the idea? Is it clear in the mind? Is it all right? Okay, ji. So I hope you got a basic idea about the glycocalyx. I told glycocalyx is of two possible types. If it is loosely arranged outermost layer, it is called slime layer. If it is tough, it is called capsule. Got the idea? Function, protection of the bacteria. Second, it hold moisture and prevent bacteria from getting dry. Third function I wrote for capsulated bacteria, it prevent itself from what? Getting engulfed by WBC. What is the meaning of capsulated bacteria? Who have capsule, non-capsulated bacteria who don't have capsule. So outermost layer is over. I hope you got a basic picture in mind. Now we are going to talk about the second layer of the bacteria, and that is what? Cell wall. Cell wall. It is the second layer of the bacteria. What is polysaccharide? What is polypeptide? Very good question, but sorry dear, it is not the right time to tell you about what is polysaccharide and polypeptide this time, because it is a chapter is there where we will talk about that. That is in biomolecule. There is a dedicated chapter for that, where you will understand what is polysaccharide, what is polypeptide. Got the idea? These are the a dedicated chapter are there. Okay, right. If you talk about polysaccharide, polysaccharide is a polymer of glucose, you can say. And polypeptide is a polymer of amino acid. It is. Then you will ask what is polymer. So I will answer all these questions when the relevant topic will come. So you will got there polysaccharide also, polypeptide also. Meanwhile, you simply, what we are saying, accept it. Ki sir told it is made up of polysaccharide and polypeptide. Sir will explain later what is polypeptide, what is polysaccharide. We will explain everything in the coming course of time. Don't worry about it. So, diagram you people not did it? Not noted this diagram? One student writing, sir, once again about, sorry, say that, show the diagram. Noted, na? So I will, in last, I will also give you the QR code. You can take a scan of the QR code. Entire board will come on you, come on your mobile. Don't worry about it. Getting the idea or not? Is it clear? So let's come on the cell wall. Otherwise, see na, if you write, another student will suffer who already noted it out. So don't worry. You just scan the QR code. You will got the things, and from there you can note it down. If you are just lagging behind. Now we are moving ahead. Give your attention. Haji, so where we was? We are on cell wall. Give your attention. So if we talk about the cell wall of the bacteria, we are just talking about bacteria, prokaryotic cell. We are not talking here about eukaryotic cell wall, plant cell wall. We are talking about bacterial cell wall. So I am writing bacteria so you don't confuse. Otherwise you will find like it is cell wall of plant. So bacterial cell wall I am writing. Or you can say prokaryotic cell wall I am writing. Got the idea? Eukaryotic cell I will tell in detail later. First prokaryotic cell, then I will say about eukaryotic cell. Right? So if we talk about bacterial cell wall, it is made up of peptidoglycan. Got the idea? It is made up of peptidoglycan. And if we talk about peptidoglycan, it is made up of course, many things in your mind. Now, you are, what is peptidoglycan? It is made up of cross-linking of NAG and NAM. Got the idea? Cross-linking of NAG and NAM. Now, what is NAG? It is a chemical compound, organic compound. Its name is N-acetyl glucosamine. And NAM stand for N-acetyl. It is a chemical compound, muramic acid. So simply you have to keep in your mind. That much detailing not required for NEET exams, or what is the structure of NAG, what is the structure of NAM, how they are cross-linking, how they are making peptidoglycan. Definitely these detailings not required for a NEET exam. We can teach it, but I told you in the very first class, we must be very centric for the exam. Your right now intention to got a very good rank in the NEET exam. And I told in very first lecture, dear children, it is very important to know what to not study. Because now world is over-informed. Over-information are there on earth about the information of material in any subject. Flood of information is there. It doesn't mean all information are useful for our exam. So smart students are those students who know what is of the relevant information for my exam. I told ki you will find a research paper of 500 and 700 page only about peptidoglycan. 5-10 people did PhD only on peptidoglycan. That is not your cup of tea. Your cup of tea, simply keep in mind कि भाई bacterial cell wall is made up of peptidoglycan. And even this NAG and NAM not written in NCERT. I said one line extra. Got the idea? Ki these peptidoglycan is made up of NAG and NAM. Got the idea? These line not written in NCERT. Earlier, old time it was there. So in old PYQ, you will find some question based on NAG and NAM. But in last 10 years, no one asked the question from these NAG, NAM. Got the idea or not? Is it? So that is the reason it is also while teaching, it is also, or while studying, it is also important to control our emotions. To teach or study that thing only who have relevancy with the paper, exam, real exam. Got the idea? Done. So it is also important. I told very first line while teaching, you must know first what to not read before knowing about what to read. See, we know many things. When we teach, you will find, oh my God, sir have how much knowledge. They are saying a great thing. But usually, dear children, these big great things not came in exam. So it is your waste of time. Got the idea? So it is very important to be very centric to the exam. So when you just write the exam after 2 years, you just find like with a smile, I am just getting full marks in exam. Sir told everything. Got the idea? That is very important to know. One student asking, sir, will tomorrow exam will be tough? Why it will be tough? What we teach in the class already up to today, we will ask in the exam only that. At the end of today's lecture, what is going to finish, we are going to ask in the exam. Nothing extra we are going to ask in the exam. Getting the idea or not? Right? Exam is online. You will got the detail. Got the idea? Exam is online. Okay? So see the thing. So what I am telling, ki this cell wall is made up of peptidoglycan, and this peptidoglycan is what? Made up of cross-linking. Made by cross-linking of NAG and NAM. So NAG stand for N-acetyl glucosamine and NAM stand for muramic acid. Got the idea? Is it clear in the mind? So this is the composition of the cell wall of the bacteria. Now NCERT wrote based on staining. Based on staining, staining means coloring by dye. We classify the bacteria into two categories. Bacteria into two categories. One bacteria is called, so bacteria I am writing and I am classifying into two categories. One is called gram-positive bacteria and another is called gram-negative bacteria. So why we are using the word gram? Because staining technique of bacteria, staining means coloring technique of bacteria is discovered by or developed by Christian Gram. C. Gram I am writing in short. Christian Gram. Got the idea? In honor of his name. Got the idea? We classify the bacteria into gram-positive and gram-staining. So gram-stain, he developed gram-stain. So he stain the bacteria, our duty is this. NCERT simply wrote gram stain, gram positive, gram negative. I am just decoding it. What is gram positive, gram negative? He stained the bacteria with crystal violet, or it is also called gelatin violet. So this is called what? Gram stain. Crystal violet and gelatin violet is called gram stain. So the bacteria who retain the stain is called gram positive, and the bacteria who not retain the stain is called gram negative. So what we are doing? We are staining the bacteria first. We are staining the bacteria first. After that, we are washing it with the alcohol. We are washing it with the alcohol. Getting the things? After washing the alcohol, who retain the stain means not washed completely. It's called gram positive. But the bacteria be stained with the gram stain, that is crystal violet or gelatin violet, and who lose the stain is called gram negative. Got the idea? So who get stained by gram stain is called gram positive bacteria. Who not get stained by gram stain is called gram negative bacteria. Got the idea? I will tell you about staining technique more in tomorrow class. Physics are already came, so definitely I am just going to stop the today lecture here only. About the gram staining, I will say something more in the tomorrow class as well. Getting the things or not? But meanwhile, you keep one thing in your mind only. That is what? There was a scientist whose name was Christian Gram, who developed a technique to color the bacteria. Who get colored by that particular stain developed by gram is called gram positive. Who not get colored is called gram negative. So is class is there tomorrow? No. Tomorrow is your exam and you will got the procedure also. Don't worry. People send on your group how to enroll with the exam, how to log in the applications. Everything they will tell you. Don't worry about it. You don't bothered about it. It's our duty to take care how to enroll, how to write the exam. And it is an online exam, not an offline exam. I am sending the QR code. Okay, ji, bye-bye. So, did you understand? NCERT based exam, yeah, related to NCERT only. What we told till today, we will ask in exam. Got the idea? And we are saying NCERT, so you just go with our notes. Nothing outside will come in the paper. Okay, ji. Bye-bye. Have a good day to all of you. Thank you very much.